Okay, hello everybody. Anyways, we'll get started on time. Hello, it's a busy day, so we'll try and start on time and get everything running on time, hopefully. My name is Aoife, and I'm one of the lecturers here in genetics. So I'm going to tell you a little bit generally about genetics here, and then give you one specific example of where we can use the genetical approach and specifically an evolutionary approach to try and understand something important and interesting. So this is our department. This is what it looked like just before it was occupied. So now there are tables and chairs on that ground floor, and it's where all our students have their lunch, and there's tea and coffee in the morning, which sounds like a trivial detail, but actually it's quite an important thing for creating the atmosphere in the department where the students kind of, I suppose, feel at home, and they're kind of well respected and well nurtured within the department. So genetics is a way of looking at biology. It's a broad approach to biology. And in this picture here, I have the genome at the center, because we really start at the genome and we're thinking at the DNA level, at the level of genes, but then we're trying to interpret that and make sense of that in lots of different ways in all kinds of different systems. So within the department, we have people who are looking at plant genetics, so looking at the process of development in plants. How does a plant grow from a seed? How does flower development initiate? But also, so this is important generally in terms of understanding biology, understanding these principles, but also, and we rely on plants a lot, right, in terms of agriculture and food. So it can be also important and implied in that kind of way. We also have a lot of work on domesticated animals in terms of understanding the process of domestication, because this was artificial selection done by humans, where we selected animals for suitability and ease of farming. And this is a place where you can kind of see that genetics and archaeology kind of come together as well, because you'll be using some archaeological information from time to time and combining that with genetic information and in terms of genetic diversity and geographic patterns of diversity. And we do the same thing as well with human population diversity, looking at you can trace genetically, you can trace the migration of people throughout the globe, starting from Africa about 70,000 years ago. They are also there's a very good research group looking at apoptosis, which is a special kind of cell death. So there's two kinds of cell death. One is necrosis, and the other is apoptosis. Necrosis happens sometimes if you have an infection, and basically the cell just bursts and leaks its contents into the tissue, and you get inflammation and all kinds of messy stuff. Apoptosis is when a cell dies and gets packaged into these tiny little blebs, and it dies in a very controlled way. And this is a really important process in development. And this is kind of a paradox, because why should cells dying be important in how you grow? But it is. And because the right cells have to die at the right time. And one little example, kind of trivial example, is at some stage all of us had webbed fingers in the womb, but this skin in between your fingers died, not in a random haphazard way, but in a controlled way by cell death. And cell death is really important throughout your life, not just in development, because all of your cells have to die after a certain point. Otherwise, you would keep kind of getting bigger and into an irregular shape, I suppose. And when cell death doesn't happen, so when this control mechanism breaks down, you have uncontrolled cell proliferation, which is otherwise known as cancer. Right? Cancer is uncontrolled cell growth. So understanding these basic principles that regulate cells and cells dying at the right time is actually an avenue into understanding cancer at a very fundamental level. And so we have this kind of research, but also other kinds of research into cancer genetics, trying to understand what's going on there. We also have research into neurogenetics. So this is a fly embryo, Drosophila embryo, and colored here, this is taken down the microscope, but this coloring is showing you different kinds of neurons within the fly. And the way your nervous system grows is not random. It's not unique to everybody. It's in fact very much the same in everybody, because it's genetically controlled. And so we can understand from a genetical approach, we can understand how is your brain wired up, how is your nervous system wired up, what happens when that goes slightly wrong. Can we understand some neuropsychiatric conditions from a genetical point of view? And there are a lot of insights to be gained there. And there's several of us working on this kind of thing. So I have colleagues working on it very much in the lab, doing experiments on neural growth, and I'm also collaborating with them from an evolutionary perspective, trying to understand more about the genetics of some of these conditions. We have genetics of bacteria as well, because so not only are they interesting in their own right, bacteria, there's more bacterial cells in what you call your body than there are human cells, and so we're living a very close relationship with bacteria, and sometimes they make us sick as well. So there's pathogenic bacteria, so they're important like that in their own right to understand them, but also we can do experiments on bacteria that we couldn't possibly do on other organisms, so they're very useful for understanding these general principles of genetics. And also within the department we have one of the leading groups in the world on genetics of eye diseases. So this picture here shows you normal vision, and this one here shows you somebody who has quite advanced retinitis pigmentosa, which is the most common form of inherited blindness. And in this case, people are born with normal sight and they lose it progressively, but starting reasonably young, so it might be as early as their 30s. And so, but this is genetically controlled, and in the genetics department here, they've been working on this for very many years, and they have gone through, I suppose, the whole process of understanding something, starting out with there is the condition, we know it's genetic because it runs in families, but we don't know how it's genetic, we don't know what the gene is. So they started studying it, they discovered what gene was involved, they discovered why, how the disruptions or the mutations in that gene actually result in this disease, and now they're in the process of developing therapies for this. So it's really a really interesting, amazing project that's gone from essentially minimal knowledge they've gone through understanding all the different aspects of this. And in this, the genome is at the center because we're understanding everything from a genetic point of view. So that's my broad, sweeping general description, and now I'm just going to talk a bit about one specific area where you can use a genetic and an evolutionary approach to understand things. And because I was told I should make a catchy title, I went with Attack of the Killer Mutant Virus, kind of as a joke at first actually, and then I went with it, and kind of then inspired by my initially joke title, I've got a kind of a B-movie theme going on here. But basically we're going to we'll talk about how you can use genetics and an evolutionary approach to understand HIV. So initially, when HIV was first, it was gradually discovered. So there was this strange illness in the 1980s that people were dying from and nobody understood what it was. So, I mean, the amount of knowledge they had is kind of summarized up in this old poster here, but people didn't know what HIV was. And they had no clue, and the knowledge the progress went from almost nothing to quickly recognizing that actually this is a virus and that is attacking immune cells in the human body, and that's how it causes disease. So we started out with something we knew very little about, and taking a scientific viewpoint, we discovered that this was a virus. And here, this is a picture of a cartoon version of HIV, and this is a human cell. And what the virus does is, it comes along, it doesn't collide randomly and just explode its contents. There's a specific reaction here. These are kind of representing proteins on the surface of the human cell and proteins on the outside of the virus. There's a binding of these proteins here, which allows then the virus to empty its contents into the human cell, and the virus genome, it's actually RNA, gets made into DNA and put inside the human genome. That's how, and then this part of the virus DNA that gets put into the human genome of the patient, it gets integrated there, and forever after that is a virus-making cell, right? So this DNA contains the instructions to make more viruses, and so this is this new virus that wasn't understood. And so the kind of thing you might ask then is, like, so how does HIV cause AIDS? What is this? How does this syndrome come about? And it is because the cells that HIV enters are cells of your immune system. They're T cells, CD4 T cells. And so what you see is, if you look over time, so the blue here is the count of those cells in the body, and the green is the count of the virus. And you see, progressively this blue line is going down. So those cells are essentially being killed by the virus, and they're immune cells, which is why the syndrome is an immunodeficiency. So the virus goes rapidly up. When you call it AIDS, when it gets to a certain critical threshold in terms of the amount of virus and the amount of CD4 cells that are left. So that is what AIDS is. And yesterday was World, I don't know if they called it HIV Day or World AIDS Day. And so this is a huge problem, and it's still a problem, by the way. So apparently the rates of HIV infection are increasing again among young people in Ireland, young men, in the 18 to 25 age bracket. So use a condom. This is an avoidable virus. So yeah, well, I mean, so it's a huge problem. If you look around the world, so here the darker the red is, the higher the rates of infection. And you can see in sub-Saharan Africa, it's a huge problem, and it's a problem doubled and tripled over because it's common there, and there's also less easy access to the medication as well. So this is an important virus. It's a big problem globally and locally as well. So if you're thinking about HIV, what are the million-dollar questions? So we've got the where, why, how. Where did it come from? Why can't we make a vaccine? And how come some people are resistant and some people are, which you might not know. So in terms of where HIV came from, this was a big mystery. So there was a mysterious virus and it seemed to come out of nowhere. And so it came from outer space, was as good a theory as any at one point, or from beneath the sea. So I'm obviously messing here, so we can see what did the papers say. And so the papers say AIDS is made in the lab shock, or Hitler created AIDS. So this is not the British Sun newspaper. This is an American magazine, and the front cover of this particular issue led with the story "Stuffed Bunny Makes 3 Girls Pregnant." So obviously a huge scientific authority. But so if we can. You can have a hypothesis if you like, stuffed bunny makes 3 girls pregnant, but you better, if you want to approach that scientifically, you can test this. So, we've got these ideas, where did HIV come from? We can try and take a scientific approach to address these. So, specifically, we can take an evolutionary approach. So, we can think about then, if you think about family relationships, you're related to your siblings and your parents more closely than you are related to your third cousin. And the same is true for species. So more species that are shared an ancestor more recently are more similar at the DNA level than species that shared an ancestor a longer time ago. So if you share a grandparent, you're more closely related than if you share a great-great-great-great-grandparent, okay? So the same logic applies. So we see that genes are passed from parent to child, or from virus to virus, offspring virus, and they're more similar between close relatives. So so if we think about this in the family tree, you're familiar with the family tree, so you and your sibling are more similar to each other than either of you is to your cousin, okay? So we take this same approach, the same logic, but just add more time, and you're talking about evolution and species. And so if you're talking about human and chimp, share more genes in common than either of them does to mouse, because human and chimp are the closest relatives, and mouse is a more distant relative. So that's probably familiar to you already. But you can apply this to anywhere in biology, and so you can apply it to viruses. And so if you want to ask, where did HIV come from, you can rephrase this question as, what are the relatives of HIV? So we've got our family tree, and we want to know what are these other parts of the tree. And so we can do this looking at the genome in the virus. So it turns out that there are other viruses that are quite like HIV, and they're found in monkeys. And so by extension, the human immunodeficiency virus, they found it in monkeys, so they call it simian immunodeficiency virus. But actually, it doesn't cause immunodeficiency syndrome in monkeys. So that's something to bear in mind, and we'll come back to. And this is an ape, not a monkey, of course, but it's also found in apes. So what you see when you look at the virus is found in all of these different primates, and it's found very commonly. So most adults are infected, and it is usually not making them sick. So as far as we can tell, it's not making them sick. And it's really common, and what has happened is, on at least 3 separate occasions, humans have been infected from primates. And so HIV-1 is the version of HIV that is spread most around the world. It's called HIV-1. There's HIV-2 as well, which is another slightly different version. So it's a separate infection that happened from the sooty mangabey monkey. So HIV-1 came from chimpanzee into humans. HIV-2 came from sooty mangabey into humans. And we can see this because when you look at the DNA, that's what's most similar. You see that it's really, really similar to the sooty mangabey virus. And HIV-1 came from chimps at least twice as well. And then there are possibly other examples. So what happened here is it happened first in Africa and then it spread. So it happened where these primates are living. And their chimpanzees and other primates are hunted for meat. And hunting is by definition a messy process, just slaughtering an animal. And every now and again people might cut themselves. There's lots of plausible ways how you might get some mixing of the blood. So what we think happened is actually this kind of infection would have passed quite a number of times, and it's only on a few relatively rare occasions that it established itself as a human infection that could spread from human to human. So if you think of the analogy with the bird flu virus, so you might have, sometimes you'll hear of one case of, say, one man who works with poultry getting bird flu and getting very sick, but there's a different stage and a different evolutionary step or genetic change for that virus to be able to spread from human to human. And this happened with HIV. So it was a monkey virus, primate virus that we got, and it did successfully start spreading from human to human. And another aspect that was important in the establishment of HIV as a human virus was the fact that colonial cities were established in Africa. So they suddenly had this great intensity of population, so a lot of people meeting each other, so they have more opportunity for spreading the virus. So if you imagine, so if one man gets infected but he doesn't live in a very big village, there's a small number of people who may catch it off him if it's capable of being spread. But once you've got cities, then the opportunity is drastically increased. So the timing of HIV, you can, with this genetic and evolutionary approach, you can also get a measure of time, and the timing of when HIV started really spreading coincides with when cities in Africa started getting really big as well. So you can see a lot of things came together here. So as I mentioned, there's a serious point to be made here, obviously. So these viruses don't make the monkeys sick, okay? So there is this tiny difference between, so everybody knows probably at this point, you know, human and chimp are 99 or 98% identical. This has become common knowledge. You can even get a t-shirt that says 99% chimp, and everybody knows what you're referring to. But somewhere in that tiny difference is the big difference, because somewhere in that tiny difference is the difference that makes us quite so different from chimpanzees. But also we have examples like this, where a very, very similar virus is causing a devastating, fatal syndrome in humans, and the monkeys are living with it, a bit like the way we live with the cold sore virus, right? So probably more than half people in this room get cold sores from time to time, and that means you have a virus which integrates its genome into your DNA the same way that HIV does, in that it's once you've got it forever. But it's no big deal, right? It's a bit of a nuisance if you get a cold sore from time to time, but it doesn't really make you sick. Chickenpox does that as well. So they are actually both types of herpes virus. And then chickenpox, once you've got chickenpox, you have that virus for life. But a relapse is relatively rare. When it occurs, it's called shingles. But once you have chickenpox, you have chickenpox. So we can sometimes live with viruses, and monkeys live with this virus, but we can't. It makes us really sick. So somewhere here is a very important difference that we don't actually understand yet, but there's perhaps an avenue for understanding things better there. So that's where HIV came from. So another interesting question is, why can't we make a vaccine? Nobody has succeeded in making a vaccine to HIV yet, despite the fact that there's been a lot of effort and a lot of money put into this. And part of the reason comes down to the genetics of the virus and the way it evolves. So what vaccines do, essentially, is they're educating your immune system. They're exposing it to something in a non-pathogenic form, so that it can then make antibodies that will recognize that next time and quickly seek and destroy, in a sense. And so One classic vaccine is the rabies virus vaccine, and rabies evolves very, very slowly, and the vaccine that was made by Pasteur still works, right? So that vaccine still works. They didn't need to make a new one. Influenza, flu, evolves faster than that. So we need a new vaccine every year or two. So there's a big, huge international scientific effort which is monitoring the virus that's circulating in the populations, anticipating what's going to be the dominant virus in 6 months' time, and rapidly manufacturing that vaccine in labs and distributing it for vaccination programs. So this is a huge thing that's going on all the time. I mean, it's an amazing coming together of all different kinds of science. So you've got evolutionary genetics, where they're anticipating what's going to be the most common virus, then the immunogenetics and immunology, where they're growing up these vaccines and then distributing them. So we've got rabies changes so slowly that Pasteur's vaccine will still be effective. Influenza changes faster, so we need a new one every year or two. And the problem, or one of the problems with HIV, is that it is evolving extremely rapidly. And basically, it evolves faster than you can make a vaccine. If you picked a point in time and you make a vaccine for this kind of, say, blue version of the virus, by the time you've got it ready, the virus is going to be changed. And it's not going to be, the vaccine is going to be ineffective. And this happens so quickly. It happens within the patient as well. Basically, the virus keeps changing that your immune system can never catch up, and that's part of why it is such a bad infection. So these vaccines are essentially going out of date faster than they can be made, and that is it's not the only problem. There are other kinds of vaccine approaches that might be plausible, but so far none of these has worked for HIV, and this is one of the major reasons why. So then, the last question we have for today about HIV is, how come some people are resistant? And this was discovered completely by accident in Paris, if I recall correctly, where there was a doctor who was researching HIV, and he discovered that there were some people who were they had basically what you might call a risky lifestyle. So they were drug users and they're sharing needles with infected people, but somehow were never becoming infected themselves. So this doctor was monitoring people. And so he tried to figure out why there were some people who apparently were not getting infected at all, despite what looked like repeated exposure to the virus. And so, as I mentioned already, the virus comes along and it doesn't just explode into the cell. There's a specific binding going on here with proteins on the surface of the human cell and on the surface of the virus. And it turns out there are some people who don't have one of these proteins, or they have a very radically different version. And so in that case, because this binding reaction can't happen, HIV can't enter the cell. And this mutation, this difference, is called CCR5 delta 32, because that's chemokine receptor 5, that has delta stands for deletion, and 32 is the number of bases in the DNA. So there's 32 bases that have been deleted from the middle of this gene, and this makes a difference between HIV being able to bind and enter or not. And this shows you here the dark red is the highest, down to the pale blue is the lowest. This shows you the frequency of this version of that gene around this part of the world, and you can see it's highest here, and it kind of radiates out. It's kind of dissipating out from there. And so this version of the gene, this mutant version of the gene, predates HIV. So it's older in human populations than the HIV infection. So it's not there in order to make people resistant. It is not a response. It's not selection for resistance. It's a lucky coincidence for some people. And it could be, we don't really know actually, so it could be just one of those things, like blood group. Some people in this room are going to be A, O, AB, and B. They're different. That's a genetic difference, but we don't know any. There's no advantage or disadvantage to being one blood group or another. Okay, so we get differences like this. We get genetic differences that are in the population, and in some parts of the world, A is really common. In some parts of the world, A is really rare. So we get these geographic differences, but they don't mean anything. So that could be what's going on here. But there are other ideas as well that it's actually to do with a different virus, not HIV, but it could have been either smallpox or plague, which are old viruses, which were really widely spread. So a lot of people were exposed to them, so there would have been a significant advantage to being resistant. So we don't quite know, but there's still lots of interest there and lots to understand. And that's the end of my talk. That's me. Paul Sharp gave me some of the pictures, and I'd be happy to take any questions. Oh, and you're supposed to leave by this exit. I was supposed to tell you that. Anybody have any questions? Yeah? The virus won't establish an infection in them, because this is very unlikely. If you discovered you had this version of the gene, don't consider it a carte blanche to go ahead and do what you like. Yep. That what? Quite possibly. I mean, I don't know that, but yeah.